All right, thank you everyone for being here. It's been a very long day. Um, I'm actually a little bit let down that there was no thunder because I'm going to be talking about one of the things that I find actually genuinely kind of terrifying about the Great Lakes. Hello everyone, my name is Ruben Jimenez, and this summer I had the pleasure of working with Shay Keretz and Anna Boegehold over the course of this summer as a CIGLR Summer Fellow on the Investigating Quagga Mussel Growth and Respiration Using Lab Experiments project. And first, I'd like to give some quick acknowledgements to Margaret Throckmorton, Mary Ogdahl, Ashley Elgin, Casey Godwin, Paul Glyshaw, Tijo O'Brien, the rest of the DEB team, and my fellow CIGLR Summer Fellows, and my friends that are watching me right now. Hello. If you can see me, hello, hi guys. Okay, so a little bit about me. My name is Ruben Jimenez. Hello, my pronouns are he, him, they, them, and I am a rising junior at the University of Michigan Dearborn. My chosen major is environmental science with a concentration in ecosystem sciences, and I am from Detroit. I was originally a sociology student until I volunteered on the Nature Center on my school's campus in that bottom right photo, which just generally got me invested in environmental science. And that summer I decided to, well, I was able to get an internship with the Detroit Zoo, which got my foot in the door in terms of aquatic ecology. Top right photo is me grabbing a net and putting it into the Detroit River to filter phytoplankton, which is where I learned about the term dreissenids. But I was kind of like, I don't see myself working with dreissenids, but here we are now. Yeah. Okay, so dreissenids, what are they? There are two specific species that exist within the Great Lakes. They are invasive, introduced from Europe, and they are Dreissena polymorpha, zebra mussels, and Dreissena rostriformis buchensis, otherwise known as quagga mussels. I will refer to them as quagga mussels for the rest because it makes it a little bit quicker. Quagga mussels can survive at depths of 120 meters, while zebra mussels can only live at depths of 30 meters. And they are both invasive bivalves that have significantly altered the ecosystem dynamics of the Great Lakes and structural dynamics of the Great Lakes. So 3 of the major kind of impacts that I found dreissenids have on the Great Lakes ecosystem as a whole are fouling, which is where quagga mussel populations have such high population densities and they attach themselves onto either ecological structures such as boat props or water intake valves or just like rocks as well. And over time they degrade these ecological or man-made structures and just kind of deteriorate them over time. They're also very good at filtering because they're voracious phytoplankton consumers. So they make very clear water, which is great if you live on the shores of the Great Lakes, but bad if you are a larval fish or if you are a competing native mussel or fish trying to find food in the Great Lakes. Along with that, they also help, well, not necessarily help, but they do this process called biofouling, which is where they also have such high densities and they attach themselves onto native mussels and also just have such dense populations that they take up native habitat that a native mussel or fish populations can have and excrete their waste on these native species. So generally we know that quagga mussels are very harmful for the Great Lakes, but there is not a ton of research that has been conducted specifically on quagga mussels themselves. We have mainly focused on zebra, well, not we, but in general, the Great Lakes community has sort of put quagga mussels on standby and focused on zebra mussels. But in this study, we wanted to build upon a previous paper, the Tyner et al. 2015 paper. They conducted a quagga mussel respiration experiment, which was pretty huge because it was the first paper/study that ever had conducted any respiration on quagga mussels specifically. They ran their experiment from 5 degrees Celsius to 20 degrees Celsius, and we kind of wanted our objective to be to build upon and just gather more information on quagga mussel physiology. And another smaller but generally also important part of my project this summer was to take this data and put it into a bioenergetics model that the DEB team is making, shout out to the DEB team, for quagga mussels in the Great Lakes and Lake Michigan specifically. Okay, so generally our methods this summer are that we collected our mussels. I was not here, Ashley Elgin collected the mussels in late March of this year, and we fed them daily with nanochloropsis, otherwise known as green algae, and just changed their water every week, made their water like every 3 to 4 days, just generally kept them very healthy. And at the beginning of the respiration experiment, we would clean the shells of the mussels with razors to remove any algae buildup and any other like organisms that were on the shells to make sure we had very nice consistent data. And here on this picture is a mini dot, but prior to the summer, I didn't know what a mini dot is, and I'm assuming you guys don't know what a mini dot is either, which is just essentially a mini probe that collects dissolved oxygen and temperature measurements in water. Over the course of the summer, Casey built these tricenetrons, shout out to Casey, which is just a mini dot that was screwed onto an 133 milliliter jar with screws and O-rings to create an airtight seal. No oxygen is getting in that thing, no oxygen is getting out of that thing. And there were mussels, obviously, but they were acclimated at 6 different temperatures for a week. We had 5 mussels per replicate, and there are 5 replicates in total, and approximately 150 quagga mussels were used for the respiration experiment in its totality. Oh, and that's just kind of the stages of building the process, and that's at the end once it has water and quagga mussels in it. And this graphic kind of just shows what I was already talking about. Oh, that's better, awesome. This graphic just generally shows what I was talking about. We would assemble the tricenetrons after placing 5 mussels per replicate, assemble them underwater, and then take them out and let them run in an incubator for 2 hours before taking the mussels out per replicate, giving those mussels each a code, getting their shell length, getting their wet weight, and then shucking them / dissecting them and placing their tissue in that upper right corner in these little weigh tins, and then putting the weigh tin in an oven for 48 hours and getting their dry weight. Getting their dry weight and then plugging that into our respiration rate formula, which is in the middle. So once we got our results, I created this nice little plot to kind of visualize our data further. So I ran an ANOVA and a Tukey postdoc test onto this graph. And as you can see, the 2 degrees Celsius, which is the white box at the very left, is pretty high above the rest of the 10 degrees Celsius and the 15 degrees Celsius, which we suspect was an equipment failure. So it is not a failure of our methods. Our methodologies is nice and sound. It was just issues with the mini dot batteries. And over the course of the respiration experiment at 30 degrees Celsius, 2 of our experimental mussels actually died during the run of the experiment because, as you can see, at 25 degrees Celsius, there is a decrease in respiration rate for quagga mussels. Because quagga mussels have a critical rate of 25 degrees Celsius, anything beyond that, and they are incredibly stressed by their environment. At 30 degrees Celsius, quagga mussels can only live up to a week, and considering that we acclimated our mussels for a week, it is not surprising that we had some like pass away during the experiment. And our p-value was 0.0131, yeah, which is smaller than 0.05, so we could reject the null hypothesis that quagga mussel respiration is not affected by temperature, which lines up with Tyner's study, which is awesome. Okay, so to compare to Tyner's study, there were obviously some similarities. Both Tyner's study and our study showed that quagga mussel respiration rate is affected by temperature based off of his p-value. Oh, that's weird. Okay, my slide didn't properly, but the Tyner's study had a p-value of less than 0.01, and they were also able to reject the null hypothesis. And there were definitely a few differences in our methodology and the way we conducted our experiment. Tyner's study only acclimated their mussels for 3 hours, whereas me and Shay let our mussels acclimate for a week. And Tyner's study only fed the mussels 3 times a week, whereas we fed them every day. All right. And a little extra point about that is that prior to each respiration experiment, we starved the mussels for 2 to 3 days. Okay, so that was a lot. I threw a lot at you guys. To summarize, 25 degrees Celsius is generally the critical point at which quagga mussel respiration rate starts to decline. Anything beyond that, and quagga mussels are not doing so great. And secondly, but also very importantly, which is the reason this study was conducted in the first place, it fills a knowledge gap. We got a lot of good data out of this, and we plan to put that data into a bioenergetics model that the DEB team is working on for Lake Michigan. And some personal recommendations for further research are that we definitely need to rerun some of the respiration, So we definitely need to rerun some of the respiration experiments from the equipment era with 2 degrees Celsius. So we'll likely run any further experiments above 5 degrees Celsius to avoid any issues. And this last bullet point is more of like a personal thing I'd like to see in the general scientific community, not something I expect from the DEB team or Shay, because it's such a large undertaking. It could definitely be its own project, but it'd be very cool if someone was able to create an intensive cross-referencing of quagga mussel population densities across Lake Michigan to just kind of gather whether they are having a significant impact on hypoxia in the Great Lakes. Awesome, okay, so this is a slide that I'm actually pretty proud about. Generally, I learned a lot from this program. Going into college, I thought it was high school part 2, and I learned that wasn't the case, and I went into this project thinking grad school is undergrad part 2, which it is not. It is just not. If I can capitalize / create one sentence to aptly describe what I learned as a CIGLR summer fellow, it is that research takes a lot of dedication, and I generally was able to put my foot in the door in terms of aquatic ecology research and realize that a research-focused graduate school program is the path I want to go down. I love doing research, as stressful as it is. It is very rewarding to see where science and where, I don't know, just like being able to work with so many different people from all different places across the globe, and just all different, like even with the summer fellows, I was able to gather a lot of information and insight from all these different types of places and people. Alongside that, I got a lot of hands-on experience with direct and without direct supervision of Shay. So I was just generally able to realize how responsible and how capable I was in a lab setting without like cracking under the pressure. I was able to conduct a lot of mussel husbandry. I was able to build an aquaculture system, maintain that system, and just generally get some experience with experimental design, which, I don't know, it's pretty awesome. All right, so thank you. Questions? Yeah. Yeah.
Great job, Ruben. Thank you so much. Okay, questions.
Yes.
Great presentation, Ruben. I really liked your dicenedron figure.
Thank you.
Yeah, I really like that. But I had a question of why you have to apply heat to mussels, like post-experiment. Like, what does that tell you, rather than looking at raw mussels?
Wait, what do you mean?
Like, you had like the tins or dehydrate. Yeah. So.
Oh, okay. So each tin was like, okay, so that's like 5 controls, and then generally like 25 tins, because we use 25 mussels per like temperature. And each tin was like this mussel. I can actually just walk over, but like say this mussel was mussel number 1, that tin right there would be mussel tin number 1. So each tin was assigned per mussel, and each little aluminum tin was measured beforehand, and then after getting the dry weight was the mussel tissue within that tin. We would just subtract it to then figure out how much tissue, the dry weight tissue overall. Mm-hmm. Yeah.
This is a great presentation and amazing work over the summer. I really enjoyed learning about your methodology. I thought that was really interesting. And you spoke about this a little bit towards the end of your slide, which I also really enjoyed, just what you got out of this and what you enjoyed about this experience. And it's really cool to hear that this has inspired you to go down a path of research and academic work. And I was wondering if there's any specific experience that really like awoken that or made you feel really excited about this.
I think, ooh, that's a good question. I think it was when, shout out to Shay, but whenever you'd get excited and be like, woo, like the mussel died, I'd be like, I was like, okay, but why are you excited? And then you'd explain it, and I was like, that's actually really cool. That's awesome. Yeah. Oh, man. I think in general, just getting to conduct, like with my hands, just because I did chem lab, like I took a general chemistry course, but it was more of like supervised, like one-off projects. And it always felt like anyone could have done that job. Whereas when I was a summer fellow here, it felt like, I mean, I'm sure someone else could have gotten the position, but it definitely felt like my mentors, especially like Anna and Shay, you are my two mentors, but you catered to me and made sure I was comfortable with the amount of work I was doing and kind of led me up to the point where I could just handle doing things on my own and not have to think about it, which I really appreciated. It just made me feel like if I could get that experience, I'm sure it's more of like a normative and not just a, you guys were just two out of like, I don't know, 100 people. And like, I don't know if that made sense, but I just generally felt like I had enough support to kind of actually gauge my interests and explore what I wanted to do and stuff. Like if I was like, I want to clean the mussels, you'd be like, okay, what do you want to do specifically? And then you'd show me and teach me, and then I'd get to do it. And it just kind of was like, okay, I like doing this. And then if I did something, I was like, I don't want to do this. And then you'd be like, that's perfectly fine. Yeah, but I don't know if that answered your question, Abigail, but yeah.
Okay, we've got time for one more question if anyone has one.
How do you tell if a mussel is dead?
Oh, that's a really good question. Because going in, I thought like I didn't know anything about mussel like biology or physiology. So I remember looking it was literally the first day, and there's like we have like the tanks. I didn't put a photo in the slides, unfortunately, but actually maybe I did. Hold on. Yeah, that tank in the bottom right, usually that's filled up with water, and we just like put our heads in and just kind of see what they're doing. And if they're completely open, that's a sign that that mussel is dead, because if you like gently tap it, they'll immediately like shut their mouths and stuff. They only open up when they're filtering, and even then it's like a little crack. Yeah. Oh, no. I mean, sometimes we'd have to like, because there's no space. It's actually a very small incubator, but. Yeah, I'm sad to see that go. That's crazy. All right. Any other questions? Or I think
Thanks so much, Ruben. That was great. Thanks for the laughs.